tutorial slot on the schedule where right now you're scheduled to talk about your projects. I just wanted to say if after I've done with this lecture, if there are some of you who want to have more time to just talk about it or ask questions and you want to use some of the slot for that, I'm perfectly happy to do so. I'll be available, so just let Mukund or me know that, that you'd like to do that, and I'll just be around. So, okay. So today I'm going to switch to a different topic. Instead of talking about patterning, as I did yesterday, I'm going to talk about growth and the control of growth. And once again, I'm going to start with a slide that doesn't directly relate to the talk, but sort of gives a little motivation for some of the approaches. And just remind people that there's really two approaches that one can take in modeling things. In mechanistic modeling, that's modeling that's actually involving real mechanisms and not just phenomena. And so the sort of most classic mechanism that classic modeling approach that biologists tend to be used to is the one in which we build models and fit them to the data in order to find out whether the mechanisms that we think are present are actually present, right? So it's a case of testing our knowledge about the system through modeling, making predictions, seeing if the observations fit the predictions, and so on. But of course, there's this other approach to modeling, which is starting with mechanisms that you're pretty confident of, building models, exploring the models, and asking whether the mechanisms are sufficient to explain what you observe in the system, right? So this is a more exploratory approach to models. And the reason why I like emphasizing this approach to models is, of course, this enables you to at least get some hints, make some hypotheses about these why questions that I was talking about yesterday. At least you can say, if it's built this way, is that sufficient to deal with these problems that I've identified or not? And so it's a nice route to understanding and to hypothesis generation. And I just put this up because I think it's important we recognize this sort of spectrum of approaches to modeling and not criticize one form of modeling for not being the other form of modeling, because they're really very different things and they have very different purposes. Okay, and so today some of the things I'll talk about are going to fall in this category and a fair number are going to fall in that category, and I hope you'll be comfortable with both. Okay. So let's start talking about this thing I brought up yesterday about, what the organism cares about is not phenotypes, not phenomena, but performance, right? The organism cares about doing jobs that could be selected for because they're useful functions. And so I'm focused, as yesterday, on multicellular organisms that have tissues, right? And all our tissues have to deal with growth. They grow during development, right? Cells proliferate and grow to form tissues that have to be the right size and so on. And many of our tissues keep growing, not all of them, but many of them are continually turning over and maintaining a size, or in some organisms they just keep growing and the organism gets bigger and bigger. So in the context of that, we can start off by trying to enumerate what are the objectives, the performance objectives of an organism that has tissues and has to grow those tissues. Okay, and so we can begin by saying, well, one objective is you have to reach the right size for your tissues, right? They have to stop or reach a steady state at the appropriate size. And there's some amazing examples of size control throughout the animal kingdom, and we'll talk about a few of them. In addition to getting the right average size, you also have to limit the fluctuations of size. Some of our tissues fluctuate in size, like our spleen, but if your skin fluctuated in size, you'd look like one of those Shar-Pei dogs, that's and you don't, right? The skin kind of maintains just the right size all the time. You have other tissues, if they change size, they, like your brain, it would just couldn't fit in the skull, right? That sort of thing. Most of our tissues are composed of multiple cell types, and so part of the growth process involves putting the right cell types in there, right? And it's not enough just to make the right cell types. You got to make them in the right proportions, because again, this is something very highly stereotyped from one tissue to another. And in a lot of tissues, it's critical that the cells be in just the right proportions. There's, again, this issue of interoperability, right? The tissues have to be matched in size. You know, your muscles are matched to the number of neurons that drive those muscles, right? Your immune system is the right size for the job it does, and, you know, your large intestine has to fit connected to your small intestine, and so on. So there's a lot of interoperability there. And then there's regeneration. A lot of our tissues do regenerate. You know, we're obsessed with the fact that there are quite a few that don't, like our brains, but many of our tissues are actually quite good at regeneration. That's why we can have skin injuries, our livers. We can regenerate 3/4 of our liver, which is quite amazing. And so that's another task that the tissues have to do. This is a task that a lot of people don't think about quite as much, but actually is probably a very big driver of the way tissues grow. And that is that there's a finite probability of mutation in the somatic cells of your tissue with every division, right? And so the more divisions you accumulate over your lifetime, the higher the risk you have of picking up mutations that might eventually lead to loss of growth control, like cancer. Okay, and so in fact, it's well known if you just take a tissue and injure it repeatedly so that it's always regenerating, the probability of cancer goes up dramatically. No mutagen required, just increasing the number of cell cycles. So mitigating those effects. And then, of course, this is a little bit related to pattern formation, is to carry out morphogenesis. And the word morphogenesis means creation of form. So a little bit different from pattern. Pattern means creation of some regular arrangement in space, right? But morphogenesis is just creation of form, of three-dimensional structure, whatever it is. And there are lots of very complicated three-dimensional structures. Think of your lungs, the branching in your kidneys, right? The shapes in your brain that have to be generated. Okay, so these are just some of the performance objectives that tissues have to face. And then we can say, all right, well, now what are the mechanisms that tissues have to deal with this, right? What's the toolbox that's available to try to meet these objectives? And so cells have a cell cycle. Now, in almost everything I talk about, I'm going to be dealing with cells that are not nutrient limited in most of their lifetime, right? And so consequently, growth and proliferation are coupled. In the sense, the cells, when they divide, right, when you stimulate them to divide faster, they just grow faster, right? They have as many nutrients as they need. So in animals, particularly mammalian cells, you know, you essentially control one thing, which is the rate of proliferation, but that's coupled to the rate of actual physical growth as well. It's hard to decouple them. So you can control the cell cycle, which controls how fast you progress through this doubling cycle. You can use factors that tell cells to change what they do during their growth and proliferation. You can have cells stick to each other, pull on each other. You can have cells sort out spontaneously so they end up in layers. You can use stem cells, which I'll define in a moment, and lineage progression, which is what you get when you have stem cells. And also, you can regulate what happens when cells divide and make two daughter cells. You can regulate what those two daughter cells do relative to each other. You know, do they always do the same thing, or does one go on and, say, turn into one cell type, and the others maybe stay the same cell type? So that's this question of division symmetry. Okay, so what do I mean by lineage? In biology, we often use the term lineage in a historical sense to refer to the pattern of divisions between the fertilized egg and a final terminal cell in the organism. This is the lineage of the entire nematode C. elegans, pretty much. And it has what's called an invariant lineage, which means that the pattern of pedigree going from egg to final cell is conserved from one animal to the next. They all use exactly the same patterns. That's unusual. Most organisms, there's a lot more variability. And so that's one meaning of the word lineage, is the sort of historical pedigree of a cell. But the other meaning of lineage, and the one that's more appropriate for today, is the idea that when you have a tissue that's even continually renewing, right? And so cells proliferate and divide, and you can draw these lineage diagrams. If you couple that with differentiation, so that, say, you know, these yellow cells, some of their progeny become purple cells, and some stay yellow. And then some of the purple cells, some of their progeny become orange cells, and some stay purple, right? That you can see over time, you know, in the steady state, what happens is you arrive at a condition like this, where, you know, assuming cells are also dying, right, that you reach a steady state where you have a fixed ratio of cells at different stages in this lineage progression. And so we draw it like this. And you might call this an enduring lineage, basically. It just means cells of this type can renew to make more of the same, or progress to make those. These can renew or progress, and so on. And in theory, you can make this as long as you like. Okay, so these are the sort of lineages that you find in self-renewing tissues. Okay, everyone understand what this means, right? It means it's kind of a proxy for saying that this is what's going on underneath. Not necessarily. Very often the final cell in a lineage is a terminally differentiated cell that does not divide. Okay, and so the thing that makes this reach a steady state is this usually dies at some rate. Okay, and so this now balances death. And so in a lot of our tissues, the principal cells, meaning the cells that do the physiology of the tissue, like the neurons in our brain, are non-dividing cells. The muscle cells are non-dividing cells. Right? It's this distribution of labor. The dividing cells are tasked with making the non-dividing ones that do all the work of the tissue. Okay. So defining a lineage that way enables us to give a sort of relational definition of a stem cell, which is simply a cell at the base of such a lineage. Okay. This is not the usual definition of stem cell that you'll see. The usual definition people will give you is a list of properties. It divides this way, it renews forever, and so on. And one of the things I'll argue is that most of those properties are not actually requisite properties of stem cells. They just sort of emerge out of the structure of networks. So I think a better definition is one of, you know, sort of where it sits in a lineage. And it's at the base of the lineage. Okay. All right, so now let's talk about the control of growth in the context of lineages, right? So what do I mean by the control of growth? Well, I said right at the beginning that one of the big performance objectives tissues have to deal with is how to make the right amount of tissue, right? And often you make the right amount and it seems to stop or reach some steady state that's very reliable. So why is that a hard problem? Because fundamentally, tissue growth, cell growth, is autocatalysis. One cell makes two, two makes four, et cetera. It's an exponential explosion, right? Exponential processes are hard to control. Just basically hard. I mean, if you think about a nuclear reactor, right, that's an exponential process. You think of the, you know, what's needed in order to keep a nuclear reactor from either going out of control or going cold. It's quite a lot of machinery and a lot of control goes into that because it's quite tricky. Whereas linear processes are very easy to control, right? They happen at a certain rate. You have them, you know, something decay, and that's it. They reach a nice steady state, right? So that's probably one of the biggest messages I want you to take home is that controlling an autocatalytic process is not trivial. And so just doing that, just reaching, you know, reliable steady states is not an easy thing for tissues. Okay. So let me give you an example of this. Let's assume you had a model of growth where you just had a stem cell and no other cells, no terminally differentiated cells. So this is like E. coli, right? It's going to grow and grow and grow. And let's say you wanted it to stop at a certain point, right? You wanted it to stop when you had 50 units of cells or whatever, right? So, you'd have to get it to stop at a particular time, and you'd have to get that very accurate, right? So let's take a particular, you'd have to either count time or cell cycles. It doesn't really matter which. So let's take a particular example. Here's a mouse brain. So it turns out if you pull out the brains of mice that are isogenic, so they're all genetically identical, so you've controlled for genetics. And you just pull their adult brains out and weigh them, and you measure the coefficient of variation. It's 5% from brain to brain, remarkably tightly controlled. But that brain started off from less than 10,000 cells, probably more like 1,000 cells that were allocated to be the precursors of the brain early in development. And so they had to go through however many rounds of duplication to get to this mass, right? So if you do the math, right, if you do the math, you find out that if this were just an exponential explosion that had to stop at the right point, you'd have to be accurate in your counting or in your cell cycles to within 0.1%. Oh, I can't, it's in the billions, but I don't remember the exact number. Yeah, something like that. So anyway, you can do the math. It's not very complicated. But anyway, the point is this accuracy is way better than the cell cycle. If you look at the cell cycle from one cell to the next, it can vary 15%, 20% easily, right? And yet somehow the final thing that's produced comes out with this, very tiny variance. So, that's a problem, right? How do you achieve that? And that's in a tissue that just kind of makes something and has to stop. In a continuously renewing tissue, right, where you're just generating cells and then some cells are dying, right? Well, the only way this will reach a steady state is if the generation rate and the death rate are exactly identical to each other, right? Any slight difference between the two, and this will either exponentially expand or exponentially contract. And so essentially, it's not a stable system. So it's because of that that people who have modeled growth, and this is certainly true in the bacterial world, right, that when you want to model a growing system, you've put in a fudge factor to make the thing come to rest at some point, because otherwise it never will. And so these are what people call these phenomenological growth laws, like logistic growth, which is motivated by the idea that, the environment has some carrying capacity so that as you get bigger, you grow slower and slower, right? And this is another form of a similar phenomenological growth law. But these are phenomenological, right, in the sense that, this is not emerging out of some actual analysis that produces that number. This is just a fudge that you put in order to make the thing work, right? And it's not, when you deal with multicellular organisms, which really don't have much in the way of nutrient limitation, right? In part because as our tissues grow, blood vessels just come in and feed them. If they grow bigger, more blood vessels come in. So it's not like E. coli in a flask where you expect they're going to run out of nutrients, right? We really don't have any good reason to use these kinds of laws, right? They're just a fudge. So the question is, what really goes on? Well, in a sense, these laws are just a way of writing feedback control, right? You're just saying that somehow the number of cells is negatively feeding back on the rate at which cells grow. And the notion that there must be some sort of feedback control, in fact, has been around for a long time in animal biology. It mostly came about from cell culture people in the 1950s who noticed that there were things that cultured cells made. If you dumped them on other culture cells, it would slow them down. And so back in the days when you could publish articles that were subtitled "A Speculative Review" and get away with it, this guy Bullock coined the term chalone, which was kind of a fusion of the words chaos and hormone, basically. So the idea was a hormone that acts to slow things down instead of boost things up. Anyway, he proposed that all tissues make some substance which feeds back on the tissue to slow down its own growth. And that's how you enable tissues to maintain a constant size or reach a constant size. And even though that was back in the 50s, it was not until the late 90s that the first such molecule was actually identified. And that molecule is known as myostatin, or GDF8, growth and differentiation factor 8. So terrible names. But I don't know if anyone's ever had Belgian blue cattle, Belgian blue steak. It's a type of cow that is bred for this double muscle, right? Well, the reason is it has a homozygous mutation in the GDF8 gene. And this is a mouse with a homozygous mutation in the GDF8 gene. And basically it has twice as much muscle as normal, right? Otherwise, the animal's relatively fine, but it's double muscled. And GDF8 is a molecule that's produced by muscle. It's a secreted factor. It's released by muscle. And it feeds back and it inhibits the ability of muscle progenitors to make muscle. So this was actually the first actual in vivo demonstration that there are such things as chalones. There have always been hints from weird experiments, which if you're interested in reading about weird experiments, read the ones about liver regeneration and parabiosis. So parabiosis is when you sew two animals together so that they share a common bloodstream. And if you take out 3/4 of the liver on one of those animals, the liver on the other animal starts growing. And both livers regenerate up to the point where the sum of the mass of the two livers equals 2 normal livers. But they're now completely asymmetric. And anyway, so that indicated that there was something conserved that was flowing around the bloodstream that was giving you a constant liver size per animal. Anyway, so these are other examples to suggest there are chalones. But it turns out that since GDF8, a number of other molecules, interestingly, many of them fall into the same protein family. A number of other molecules have been identified as chalones, as things that are negative feedback controllers of growth that come from the tissues themselves. Okay. So one of the nice things about modeling, as I said, is you can use it to ask whether your mechanisms actually work, right? Are they sufficient to explain what they're supposed to explain? And so let's just think about this chalone strategy in the simplest possible terms and see what it can do and what it can't do. So consider a tissue where you just have something that divides and you need to shut it down at the right time, right? Like that brain case, right? So what if you had negative feedback on, I'm going to use V just to represent the cell division rate. So now V is going to go down as the number of cells go up. So you might have a growth law like this, right? That the rate of proliferation of the cells, right, depends on some growth rate. And then this term, right, so here's your autocatalysis. It depends upon the number of cells, but now we're dividing by something that depends on the number of cells with some exponents, so we can have different gains of our feedback.
I think I'm being a little slow. So C prime actually is dC by dt.
Yeah.
Sorry,
Yeah.
I just wasn't with your notation.
I'm sorry. I don't know. Some people like C prime, and some like C dot, and some like dC dt. Okay, yeah. So yeah, this is, yeah. Okay, yeah. All right, this is more, that's like how a biochemist would do it as opposed to a physicist. Anyway. All right. So if you look at that, well, the first thing you see is actually this doesn't stabilize the system. The system continues to grow forever. It just doesn't grow exponentially. So for example, if you have linear feedback, then you can take the exponential system and now have it grow linearly. And if you have quadratic feedback, you can get it to grow sublinearly and so on. But you don't actually ever reach a steady state with this type of feedback by itself. Now, of course, if you have this sort of situation where this cell can die at some rate, and so I'll use D for death rate, and this means, vanishing, right? Well, then actually you can get a steady state, but it's not a very robust steady state. But by that I mean it's quite sensitive to its parameters, right? Especially around here, right? If you change the cell division rate, you can go from no cells to a lot of cells over a very short span. And remember how I said how robust the sizes of tissues were. So it's like, doesn't sound like the sort of best strategy. Now, the other thing that's a little bit funky about this strategy is, remember I said you want tissues that regenerate. And presumably you want them to regenerate as fast as possible, because when you have an injury, you want to get rid of it, right? So if you just have a system like this without any feedback, and you lower the number of cells, and you let the number of cells come back, right, they relax with a certain rate constant, right? That relaxation time is really just the inverse of the death rate here, because that's the time it takes to get back to some steady state. Well, the presence of feedback in a system like this actually doesn't speed up the return to steady state at all. In fact, it slows it down a little bit. So there's not a tremendous value to having this feedback. It doesn't give you a robust steady state. It doesn't give you fast regeneration. And as you'll see in a minute, there are other tricks you can play that can do those things very easily. So now let's imagine, instead of building tissues like you would build an E. coli colony, where you kill some of the cells. Actually, I'm trying to think. This curve, I hope I didn't mix two curves from two different situations. This would be no feedback where you've balanced, right? Yeah, and you can't take it down to 0, obviously. Yeah. But it's still going to come back with this rate constant. And this is actually going to slow the rate at which it comes back, because the speed is going to keep slowing as you get more and more cells. So we'll just see in a moment that there are other feedback strategies that can speed up the rate a great deal. But this is all sort of as a prelude to talking about what happens if we actually do lineages, right? So if instead of just thinking of things like E. coli colonies, we have stem cells that can make this transition to produce terminal cells, and then those terminal cells can die. All right, so what if we do things in that case, and now we use this same strategy of negative feedback? So it's like, okay, well, the terminal cells are now feeding back on the rate at which they're produced by stem cells. That's probably more what Bullock had in mind when he was thinking about this initial chalone hypothesis. So how well does this system perform? Well, it actually helps a little bit in terms of robustness, right? So you get a steady state, right? And the number of terminal cells you get, its dependence, say, on initial conditions or on the rate at which things divide, is slightly more robust than if you had no feedback at all, okay? But it's dependence on parameters like the rate of division or the rate of death. But it's still linearly sensitive to the initial conditions, okay? And it does regenerate faster, because now in this system, if I take away these terminal cells and I ask it to come back, of course, it speeds up for a while until it slows down when the feedback starts kicking in. So you can actually improve regeneration speed with this sort of thing. And you can do the same thing in a multi-stage lineage, right, where you have many different lineage stages. But interestingly, if you do that, if you have a terminal cell feedback on something that isn't a stem cell, but a sort of intermediate cell, then it actually provides no robustness at all. Because if you basically, because as a result of feeding back on this, these cells end up accumulating. And so you produce these cells at the same rate as if there were no feedback. Right? So essentially, in terms of the steady state behavior of this sort of feedback loop is actually kind of useless. It doesn't really do anything. And the only way that this can actually improve your regeneration speed is if most of your cells are intermediate cells and very few of them are differentiated cells. Then you can actually get that sort of twofold improvement in regeneration. So it doesn't sound like a fantastic strategy. There's a bigger problem with all of these strategies, which I sort of glossed over. And that is at this step here, these stem cells seem to have to divide in a way so that half of their progeny become the next cell type, and half of the progeny renew and become the first cell type. Because if that doesn't happen, well, then you've got your exponential explosion or exponential decline over here. And this feedback doesn't do anything for that. I mean, that has to be a fine-tuned parameter. And that's makes one very uncomfortable, right? The idea that cells, should fine-tune their behavior so that they make exactly 50% of their progeny one thing and 50% of another. Now, many biologists kind of sweep that problem under the rug by attributing to stem cells a certain magical behavior, which is called obligate asymmetric division, which is to say that every stem cell, when it divides, one of its daughters becomes one thing, and consequently the other daughter stays the same. There's all these beautiful models for how cells can pull this off. Right. There's only one problem. This is not how it actually happens. Right? I mean, it's a lovely model, but there's actually very few stem cell systems that behave this way.
When you say that it has to fine-tune, does that mean that the parameters have to fine-tune? You have parameters mu and d and so on?
Not those, but this is the symmetry or the division behavior of the stem cell has to be fine-tuned in the sense that half of the output of the stem cell has to return to being stem cell, and exactly half has to progress to the next stage. Right? Any other ratio would be unstable.
But that should be determined by the dynamics, right? You put down those equations. And that should tell you how many stem cells remain. I mean, there'll be some sort of variable for each of those blobs, right? For each of those circles.
Right. Right. So the dynamics here are just that exponential expansion with loss. And so unless the expansion and the loss are exactly balanced, the whole system is unstable.
So the numbers of the stem cells won't choose themselves?
No.
The rate of that, I see.
The rate is irrelevant. All that matters is, I mean, the rate will affect the rate at which things flow out here, but essentially, unless it's exactly balanced, you will have no steady state.
I call that number V prime. The V prime should be equal to V.
If that number is referring to the proportion that goes this way or that way. So as you'll see in a moment, we do put a number, we call it P. And P stands for the probability of return. And so what you're saying is P would have to be 1/2. And that's exactly right. Unless P is 1/2, this system has no steady state. Yes.
Is that what's really going on?
Well, so that's what I'm about to show you. Because again, most biologists don't want it to be a stochastic process. They want it to be a deterministic process that forces P to be 1/2, which is essentially to have the cells make asymmetric divisions. And it's amusing because the literature is so full of diagrams showing that this is how it happens. But in fact, we've known forever that this is not how it happens. And some of the earliest data actually go back to some of the earliest data on stem cells themselves. So this is beautiful work from the 1960s by this fellow Till in Toronto. And some of the earliest stem cell work involves taking stem cells out of animals and injecting them into other animals. So bone marrow stem cells. If you irradiate a mouse, you kill all of its bone marrow, which means it can't make any of the blood cells. But you can take marrow cells from a donor mouse and inject them in, and they will engraft and create little colonies. And in this case, the colonies are red cell-making colonies that form in the spleen of the donor mouse, of the recipient mouse. The spleen shriveled up, and then these new colonies start from those stem cells. Okay, and the way that Till showed that those colonies were actually colonies, which is amazing. He slightly irradiated the donor cells to create specific chromosome breakages, and then he showed that every one of those lumps, all the cells had exactly the same chromosome breakages, so they must have come from a single progenitor. So then he further went on, and he took each lump dissociated it into individual cells, and injected it into a new mouse, each lump into a separate mouse, and counted how many colonies came from every colony. All right? And as a result, tried to count the number of stem cells that were present in each colony by this purely operational method. And what he saw in his, these are the actual data that he published, right, is looking at the colony-forming units per spleen, right, and he's essentially looking at the number of colonies, the histogram of the number of colonies that had this or that many stem cells in it at different amounts of time. And now, normally you'd expect something like a Poisson distribution, right? But look at the distribution that he saw. This really heavy-tailed distribution where some colonies had a huge number of stem cells in them, and many colonies had 0 or 1 stem cell in them. And so he didn't stop there and say, well, what a strange heavy-tailed distribution. He actually realized that that distribution arose naturally out of something that Boris talked about a couple of days ago, which is birth-death processes, right? The stochastic processes of random birth and death. And he recognized that there are actually 3 processes that go on here, although one of them you can sort of ignore. So we tend to draw it this way, that a cell can make 2 stem cells, 0 stem cells, because both differentiate, or 1 of each. But 1 of each doesn't change the numbers of anything, so you can sort of ignore that one in the math, and you could just deal with it this way. Doesn't really matter. You can do the math either way. And the point is, what you get is you can calculate how the probability distribution arises for numbers of stem cells in clones as they go up over time. And just remember, I said that we use the term P to represent the overall fraction of a stem cell's output that goes on and becomes stem. And so that's kind of this plus half of that. And then 1 minus P would be this plus half of that. And I won't go through all the math, but I'll just show you, how easily you get heavy, very, very heavy-tailed distributions from this sort of behavior. Okay, so in fact, Till recognized that the distributions he was seeing told him that the stem cells were behaving stochastically, because that's exactly what you should see if the stem cells are behaving stochastically. Right, and in part that's because every symmetric differentiation extinguishes a clone, every symmetric renewal doubles the number of stem cells in a clone. It's exactly what Boris talked about when he was talking about genetics, the sort of stochastics of branching processes. It's really all the same math. And in fact, you can see that, for example, the mean stem cell number per colony is staying constant because on average, the divisions are producing an equal number of stem and non-stem. But the mean stem cell number per existing colony, one that hasn't extinguished itself, is going up linearly with time, which is in fact what you'd predict. And so the remaining number of colonies is going down, and it's going down as a nice hyperbola. Right? And so you see a lot of clones extinguish very, very early, and others sort of jackpot. Right? The bigger you get, the longer it takes for you to drift back down to 0. Exactly. We haven't got to the control problem. We're just saying it isn't controlled by fixing the symmetry, because the symmetry is random, right? So now how do you get it to be 50-50 when it's not deterministic? Which exponent? I think, well, so the Till distribution fits those data very well, and I think the linear growth here is consistent with the with this model, and in fact, this linear growth, this is a simulation, but this has actually been observed in other systems. The slide before. This one? These are simulations, so this is not data. Okay, but this is just using different ratios of how much is allocated to one fate versus another, just to show you how readily these distributions become heavy-tailed. But But this, this picture, this is a simulation, but exactly the same thing has been observed, for example, in the skin. Okay. So, but just to show you, in fact, this prediction that stem cells are constantly extinguishing themselves is actually correct. Later on, in some nice work from Ihor Lemishka and others involving engrafting hematopoietic stem cells into mice, they had ways of labeling each cell with a different tag, basically a retrovirus that went in at a particular point in the genome. And you can see that initially there are many different clones that engraft in the spleen, and by long term, most of them disappear, and you're just left with one. Okay, again, consistent with this branching process approach. I just thought I'd take a little diversion for a moment and say that this till distribution is reminiscent of another famous heavy-tail distribution that kind of sort of messed with biologists' heads for quite a while, which is the Lurie and Delbruck distribution. So how many people are familiar with Lurie and Delbruck distributions? A few, okay. Anyway, it has to do with what number of mutations you should expect in a population after a certain amount of growth, right? And initially, this was how people tried to measure what's the rate at which mutations are occurring, is just look at the frequency of mutations in a number of individual isolates and then take the mean, right? And the problem that Lurie had is every time he did that, he got a different mean, right? He found that he could never do enough samples to actually get that mean to converge to anything. And so Max Delbruck helped him figure out that there was a reason for that, and that is that the expected distribution of mutations in a branching process where the mutations can hit at any random point has a huge tail to it, because if a mutation hits early, it generates a very large number of mutants at the end. And so that's called the Lurie and Delbruck distribution. And because the distribution is so heavy-tailed, any sample of it, it's very hard to estimate a mean from any sample of it. And so they took a problem, which is why you couldn't measure something, and they actually showed that the problem sort of proved the model, right? That in fact mutations were just occurring randomly at any point along here, rather than say all occurring with equal probability at the end, which would have given you a Poisson distribution. So it's one of those great examples where distributions mattered in biology, and it was the recognition that the distributions that arise out of branching processes were important, that it made it possible to sort of sort out a real problem. And actually this Lurie-Delbruck problem has real relevance to biology, because we often think about mutations causing cancer, right? But the fact that we are a branching process, right, means that some of us got those mutations early on during development. And if we did, they've jackpotted essentially and filled whole tissues with the mutation. So many of us, even though we're genetically not cancer prone, we had an accident early in development, and we are now genetically cancer prone in our somatic tissues. And you can calculate what the probability of that is if you know how many divisions took place during development. Was there a question in the back? Yeah? No, just stretching. Okay, good. All right. So one of the ways in which understanding these branching dynamics is of practical significance is in another concept that emerged in the stem cell field called the transit amplifying cell. This is a term very commonly used by stem cell biologists, and it comes from this work in 1974 from Chris Potton, who was looking at the skin. So if you look down in the skin, right, here's the cornified layer, that's the thick outer, the dead layer basically, and then below that there are these cells, and at the very base there are some cells that are actually dividing. The ones down here are dividing. And you can use tricks to sort of figure out whether a cell's dividing by labeling it with a nucleotide that you can detect, either radioactive or otherwise. And then you can follow over time what happens to those cells that are dividing. Do they stay in this place? Do they give rise to cells up here? Do they get flushed out of the tissue as more divisions take place and these become dead cells and get sloughed off? What happens to them? And so what Potton observed is typically under one of these little patches. You can actually see these little hexagonal patches on your skin. Under one of those, there are about 5 or 6 dividing cells. And if you label them and follow them over time, usually all but one get flushed out. But at late times, there's always one that's remaining. And so from that, he concluded that there are 2 cell types. There's a stem cell, somewhere in the middle here, and then there are these intermediate cells that the stem cell gives rise to, and they divide for a certain fixed number of times before they rise up into the upper layer and differentiate and get shed. And that's the classic model you will read in almost every textbook. It's this idea that you have a stem cell which can divide unlimited numbers of times, that you have a transient amplifying cell which has a limited capacity to renew itself, and that's why you would control its growth, perhaps, and then a terminally differentiated cell, which doesn't divide at all. This is the sort of classic review. The problem is it's all wrong. And the reason it's all wrong is because what Potton saw is exactly what the Till distribution predicts for a single type of cell that's dividing stochastically, right? Most daughters will eventually extinguish, and some will expand up and take over. Right? And so essentially he just saw 10 years after Till the distribution that Till predicted for a single cell type, but because he was unaware of it, he said, well, there seem to be, you know, quick extinguishers and late extinguishers, and so therefore there must be 2 different cell types. And it really wasn't, right? It really wasn't until about 10 years ago that beautiful work from Jones and Simons in the skin, the very same system, demonstrated that in fact the division patterns of cells are stochastic. In this tissue, about 16% of divisions end up giving you 2 stem cells or 2 differentiated cells. And there's no predictability in terms of, you know, how one cell will behave from one division to the next. And you can build stochastic models, and you can actually fit those models to the data. I think you're going to talk about data collapse later, right? So this is actually one of these examples. You can get all these curves to collapse onto one canonical curve. It's really beautiful work that really shows that the whole system is stochastic. The work's been extended to systems like the intestine, where you have this intestinal crypt that generates all the cells of the intestine. And once again, you can do the bookkeeping and show that cells are behaving in a perfectly stochastic way. Interestingly, in the intestine, almost 80% of all divisions are symmetric. Not that asymmetric thing that everybody thought was the sort of sine qua non of a stem cell. And so then that gets us back to our control problem, right? Because we said in order for these stem lineages to maintain any sort of steady state, you must exactly balance. You must have a p of 1/2. The renewal and the progression has to be exactly balanced. But I just told you it was stochastic. So that would suggest, you know, you have to fine-tune the parameters so that this stochastic process gives you exactly that number. And that seems pretty darn unlikely, right? And so then that's where this question of feedback control comes in. And to sort of tell you a little bit about the data that support the way feedback control is actually working, let me just briefly introduce to you an experimental system. It's called the olfactory epithelium. It lives in the back of your nose. It's the reason stuff smells good, because these are the neurons up here, these blue things, that actually detect odors and send the messages to your brain. Okay, they're a little patch in the back of your nasal cavity. And there are cells at the base that proliferate and generate these cells. These cells are terminally differentiated, but they die at a certain rate, and so they're constantly being replaced. It's a part of your nervous system that's constantly regenerating. The blue cells here, and they have these little dendrites that stick out into the air, into the nasal cavity. These are supporting cells up here. We'll talk about them in a minute. Yeah, blue pokes through yellow. And up here you have mucus. Everyone's familiar with that, I'm sure. And the odor is partitioned into the mucus, and that's actually where you do all the sensing of odors. It's a really interesting structure developmentally because it starts as just a little pit in the front of your face, and it invaginates and it forms this highly convoluted structure. Okay. And it has a lineage which has a stem cell with some markers and intermediate cells. I won't call them transient amplifying cells, but they're intermediate self-renewing cells and olfactory receptor neurons, and we have markers for them. We call these INPs and ORNs. Okay. And it regenerates very nicely because it projects back to the brain. That's where the neurons attach, and if you kill that part of the brain, the neurons over here die. And when they all die, the proliferating cells start going into division, and they regenerate the neurons. And in vitro data suggests that, as in the chalone case, the neurons are producing something that inhibits their own production. And it turns out that GDF11, which is another one of these secreted molecules very closely related to myostatin or GDF8, is highly expressed in this epithelium. And if you knock it out, they make too many neurons, just like that mouse made too much muscle. And if you do the in vitro studies, you can show that GDF11 acts on this cell type, on the intermediate cell type in the lineage. Okay. So unfortunately, this diagram, right, which would be of the neurons feeding back onto the growth rate of this cell type, corresponds to something I told you earlier was the most useless possible feedback configuration. Right? I said if you feed back from a cell like this onto the rate of division of an intermediate cell, it doesn't really do any good, right? Because you slow this guy down, the numbers just build up, and it just pushes the same flux through. So it's kind of weird, right, that why should the GDF11 mechanism of action look like something that doesn't do any good? And this led us to propose a while back that, in fact, that's not what GDF11 is doing, but in fact, what GDF11 is doing is instead of feeding back on the rate of division of that cell, it's actually feeding back on this parameter, the p-value. It's affecting the choice that the cell makes in differentiation. Does it renew or does it progress? In particular, it's negative feedback in the sense that when you have a lot of these cells, it reduces the renewal rate of that and then promotes the progression. So it's kind of mixed positive and negative because it promotes the progression to the thing you have more of. Anyway, you can write that in terms of very, very simple ordinary differential equations. Here would be your negative feedback, where G is the gain of the feedback and chi2 is the number of cells of this type. And if you explore those differential equations, you see that very interesting things happen. For example, you can get systems where the steady state is extremely robust to the initial conditions or the rates of division or the rates of death, right? You can vary them all and you pretty much get the same steady state of that system. And you can also get systems that regenerate in a very rapid manner. And they do it in a way that looks very in vivo-like, which is that the precursor cells, the INPs, increase and then decrease transiently. And while they're coming down, here's a blow-up, then the differentiated cells start getting made. And that actually is what you typically see in regenerating tissues when you injure them. They don't immediately start making the terminal cells. First, they go into a phase of expanding up progenitor cells. Then they sort of push on through and make terminal cells, and their numbers go back down. And so all of that just comes right out of the ODEs. So is that really how GDF11 works? It turns out it is. I'll just gloss over this. These are experimental data in which you treat cells in culture with GDF11, and you show, you test what fraction of them go on to differentiate, which would be going up this way, as opposed to not differentiate. And you can show that as a function of GDF11, that fraction goes up. Then it flips around and goes down because GDF11 is also slowing the cell cycle. And it turns out that at higher concentrations of GDF11, the cell cycle gets so slow that you have to wait till longer to do your assay. So it's not really that it isn't, that it's not promoting their differentiation, it's just that it's taking a lot longer to get there. So it's a mixed effect of affecting both P and V, okay, which like that. Okay. Now, yesterday I talked about trade-offs. There's, Yes, as I'll show you, it arises automatically, emergently out of it. Okay, so there's some, well, in a minute I'll get to that. Okay, all right. Anyway, so there's one problem with this, which is that these two phenomena I showed you, robustness and speed, cannot be achieved simultaneously, right? The parameters that enable you to have very robust steady state preclude having this very fast regeneration, right? And the parameters that enable you to regenerate fast preclude having a robust steady state. And furthermore, this fast regeneration, it has to be fine-tuned. If you wipe everything out, you get a fast regeneration. If you only wipe part of it out, you don't get a fast regeneration. So it's not as great as it seems, right? Mm-hmm.
So in your injury experiments,
mm-hmm.
Can one say how far deep you go back in your lineage you will be?
Yes, yes, one can. And in the olfactory epithelium, these two cell types both show effects, although these ones show a very large effect, right, the ones that are close. But you also see some of these increasing. And part of that's for the reason I'm going to show you in the next slide, which is that there isn't just GDF11 in this system. There's something called activin, which is another member of the same family, and is also expressed in the epithelium. And when you take it away, you get an overproduction of these progenitor cells. Okay? Question? That's a really good question. A lot of systems have that behavior of a stem, one intermediate, and terminal. But there's no reason why it has to be that way. And there may also be resting stem cells, which sort of underlie the stem cells. So there's no a priori reason it has to be that way. Anyway, the thing about activin that's interesting is it's also produced by these cells, but it acts on that cell instead of this cell. And you can show that in culture. It's completely specific for that cell type. And so the real loop that you have in this system is a double feedback onto each of these stages. Okay? And it also affects the p-value and also the v-value of that cell. So what you get is something like this. If I put all those feedbacks in, I model them all with Hill equations, because it doesn't really matter what I model them with. All that matters is just kind of the slope around the half point. And you get, you know, ugly things like this. But the beauty of that is that if you calculate the steady state for the terminal cell, all the parameters drop out. And you get that the number of terminal cells depends essentially on the sort of intrinsic ability of stem cells to renew if there were no feedback, and the gain of the feedback, or the strength of the feedback. And everything else falls away. And so why is that? It's because this is what's called integral feedback control. All right? You have a steady state, and the only way to reach the steady state is if you get the set point. Right? And all other states are attracted to that set point. And so you get what's called perfect robustness. The only parameters that affect the steady state are, right, this parameter and the gain of the feedback itself. And you know, the reason for this is because of what you just mentioned, which is that this effectively clamps P0 at exactly 1/2. Why? Because if P0 goes above 1/2, then these cells expand exponentially, and these cells expand exponentially. So they go up and up and up, and they will not stop until P0 goes back to 1/2. Right? And so the very thing that makes these systems so hard to control, the exponential explosion, you're essentially using that to control the system. And as a result, you get this perfect robustness where the system automatically comes to 1/2. So what that's telling you is this is not some special character of stem cells, that they self-renew half the time. It's just the emergent property of a feedback loop. Okay. So I just said that. Actually, when you put in the double loop, you can also solve this problem, that you can get the system to regenerate faster from a variety of injuries. And it turns out, if you look in other systems, that it works exactly the same way. So this seems to be a sort of universal theme. As is often the case when you have integral feedback, it's very useful, and so you use it again and again. The interesting thing, which is a little counterintuitive, is that you can use the same integral feedback to solve this brain problem that I posed before, right? So if I just take that same circuit, now I'll just have one stage instead of two, just for simplicity. So a stem cell and a terminal cell. And I take away the death of the terminal cell. So now what will happen is you'll make terminal cells, and their numbers will increase until the p-value is driven down below 1/2, at which point these guys will extinguish by pushing out more and more of these terminal cells. So if you explore how that behaves, if you don't have any feedback in a system like this, well, obviously, if you have twice as many progenitors, you have twice as much brain, right? It's totally non-robust to the number of progenitors. But if you do have feedback, look at these two curves for twice as many or half as many progenitors. You make almost exactly the same size of brain. It's not perfect robustness, but it's very, very darn close to perfect robustness, simply by having negative feedback control on P. And it doesn't matter what the form is, and it doesn't matter what the gain is. The gain will just give you the value that you come to. But whether it's going to be essentially perfect is independent of all that. Okay. And of course, it solves this problem of stochasticity. Because if you ensure at the population level that this number is 1/2, you can let the cells be as stochastic as they want in their division patterns, right? There's no need to control the individual cells because the population is under control. Okay, so that's the good news. What about the bad news? Remember I said every time you have a strategy that does something really good, you have to pay the price somewhere, right? So what exactly are the costs here? So we can begin to explore that by testing some of our hypotheses about this model, right? What does it predict, and does it come out right? So for example, we already said if you knock out GDF11, you'll make more neurons, and indeed you do. So we know that prediction's met. But what happens if you knock out activin? What would this model predict? Anybody want to guess? Your nose would explode, right? Not a pretty sight, all right? If you knock out activin, that's what's restraining this p-value. So it's going to go above 1/2. It's going to expand exponentially forever. And okay, so that's not what happens. In fact, you can knock out activin and the mice live. So, and their noses don't explode. And if you look closely, you do see that the number of progenitor cells goes up. But bizarrely, the number of terminal cells and even the number of intermediate cells stays the same. So these numbers rise, and these numbers stay the same. Isn't that weird, right? If you're thinking about this as like a linear biochemical pathway or hydraulics, that should be impossible, right? You put more in here, why doesn't it flow out there? But it's not, right? These are linked exponentials. It's a little different. Okay, so what does it tell us? Well, first of all, the fact that you can increase this and have very little effect here tells us that this P1 value, even after it's under feedback control, has to be pretty high, pretty close to 1/2, so that most of the flux that's coming from here to here is coming from the renewal of these cells and not from the contribution from below. Okay? And the other thing it's telling you, since the nose doesn't explode, something else must be restraining this besides just activin, right? Otherwise, that p-value should go above 1/2. Okay. Well, there's problems with believing both of those statements. So the first thing is the condition when this is high and that's low, or not that's low, but there are very few of these because this is high. That condition is precisely the condition under which you can't regenerate fast. Because essentially these are already sort of near their capacity and it can't go up much more. And so you've sort of thrown out one of your important objectives. And the second thing is by putting any other feedback on this parameter, you throw away that perfect robustness. The integral feedback control goes to hell, right? You can't do it anymore. So that suggests that we're fundamentally missing a piece of this system, right? We obviously don't have the whole thing. So what could we be missing? So it turns out there's long been evidence for positive feedback factors that affect these p-values, right? Things that have the opposite effect, increase the probability that a stem cell will renew itself. In the olfactory epithelium, those are FGFs, fibroblast growth factors, different growth factor family. But this is a plot, essentially, of how many of those intermediate cells remain intermediate cells after one division, as opposed to progressing on to becoming neurons. And in the presence of FGF, you can increase that number dramatically. So you can affect these p-values. And we know FGFs are present in the system, and actually there are some FGFs, if you mutate them, you lose the olfactory epithelium entirely. So obviously they're important, right? So how are we going to put them in? Well, one interesting thing is, if you take a system like this, where I said there was this trade-off between you could be robust but not fast, or fast but not robust, if you just put that positive feedback in, you can actually break that coupling. You can get cases that are both very robust and quite fast, simply by having a double positive and negative feedback. Now this seems kind of weird, right? Why would you have two completely antagonistic things coming from the same cell onto the same cell? But of course, their parameters are different, right? The cell can be more sensitive to one over one range, and then the other one will kick in at a different regime of cell number, right? And so that's exactly what's happening here, is these are ones in which first positive feedback kicks in at the lowest values of these, and then negative feedback kicks in later. This is if you delete the positive feedback loop and you just explore all of parameter space, and you say, can this system be fast and robust at the same time? And this is how fast it is, this is how robust it is, and they're mutually exclusive. Now you do the same thing with the positive feedback loop here. Again, explore all of parameter space, and you find that in addition to these cases, there are these orange cases out here, which are new, where you can be both fast, pretty fast, not as super fast as this, but pretty fast and robust. Sorry? Oh, sorry. If you're colorblind, there are dots here. Okay, sorry. So where do those cases come from? Why are they colored orange? Because they're all the bistable cases. And that's one of the interesting things that happens when you mix the positive and negative feedback, is you actually create bistability, so that the terminal cell numbers have two possible steady states they can reach. And one of the reasons you can regenerate quickly here is because you can essentially toggle the system. It can be in the low state until you take away both feedbacks. It jumps into the high state, stays there as the positive feedback feeds it, and then the negative feedback comes in and bangs it back down. Okay, so that just emerges spontaneously out of that. It essentially gives you a high gain feedback, ultra-sensitive feedback. There's a sudden effect of the feedback as it kicks in. Now, of course, everything comes at a price. And what engineers know is high feedback gains have a certain price that's very common, which is instability, what's called high gain instability. And these systems show this tremendously. This is a case in which you suddenly remove half the terminal cells and you see what happens. And if the feedback gain is 1, it's linear feedback, then you see some damped oscillation. If you make the gains higher, as you might have due to this mixed positive-negative feedback, you see the oscillations get worse. And in fact, once you hit about n equals 4, you get a point where you get sustained oscillations, and the system will never go back to a steady state, in theory. Right? This is a useful way of looking at disturbance frequencies versus the sensitivity. This is that sensitivity number I talked about yesterday. So is there a way to get around that? So I've only been talking about feedback that goes from one cell back to a previous cell, affecting this p-value, right? It turns out if you introduce a branch into the lineage, right, that is, allow the stem cell to make another cell type, or even die, but let it make another cell type that's used for some other purpose. And instead of just feeding back here, you're actually feeding back positively on this cell. You're saying, let's divert our lineage over to here instead of there and there. You can actually make all this instability go away. You can take all of these oscillations and get the opposite to happen, which is the stronger your gain, the more abrupt your feedback, basically the fewer oscillations you have. And you have really perfect dynamic control out of that. And it turns out that's just what the olfactory epithelium does. the stem cell has an alternate fate, which is to produce that supporting cell that was at the top next to the blue guys, called a sustentacular cell. And activin has exactly the predicted effect of driving the stem cell down the sustentacular cell pathway. Okay? And the mutants, if you look at the levels of sustentacular cells in these mutants, they're completely consistent with that model. Oh, and here's an example of how you can get excessive numbers of stem cells by mutating some of these factors. Okay. Now, there's another price you pay for using bistability to achieve ultrasensitivity, and that's hysteresis. Does anyone not know what hysteresis is? Okay, good. Everybody knows what hysteresis is, right? The fact that where you go up the curve and where you come down the curve occur at different places, right? So you might expect that as a result of using a hysteretic system, you might actually find some examples where things get stuck in one state and not another. And it turns out that there's great clinical examples of just this sort of behavior. Hypertrophic scarring, cirrhosis, myeloproliferative diseases, and one of my favorites is psoriasis. So I don't know how many people have familiarity with psoriasis. It's a nasty skin disease, right? It causes these nasty patches. But basically, these are locations where stem cells are going crazy in the skin and producing very large numbers of themselves. Essentially, you're in a high state over here, triggered initially by some immune insult that's still not very well understood. But long after the immune insult goes away, months and months later, these things persist. And the stem cells are still dividing to produce large numbers of themselves, and as a result, much less terminal material. And that's why they look so red, because the skin's very thin. And the only effective treatment for psoriasis right now is just to randomly kill stem cells. That's essentially the treatment, is you use ultraviolet light and a sensitizer. And if you can get below a threshold number of stem cells, the lesion goes away permanently. And that, of course, is exactly what the model predicts, right? You have a hysteresis, and you're stuck in what's effectively irreversible position on the high part of the bistable curve. The converse of being stuck in a high state is also being stuck in a low state. And this is another phenomenon you see all the time in development. When you mutate or take away a little bit of the function of something, you'll reach some threshold below which some organ will simply vanish. Right? It just won't develop at all. And that's called agenesis. And that's a very common form of birth defects, is agenesis. Right? And that's exactly, again, what the model predicts, is that varying any of a number of things, like initial stem cell number, or the strength of the feedback, or any external signal, can give you a situation where growth will never take off, because you can't get off that lower curve, and then you get above that, and then suddenly growth ignites, in a sense, like a burning match. And same way you can go down in the other direction. And in fact, the olfactory epithelium shows just that sort of behavior. There's a mutant called FOXG1, which plays a role in GDF11 expression and signaling. And when you take it away, it's like you have way too much GDF11 activity, and the olfactory epithelium just vanishes. You get none whatsoever. But interestingly, you can titrate that in by taking away doses of GDF11, 1 copy, 2 copies. And you can see abruptly the epithelium comes back when you've just taken away 1 copy of GDF11. Doesn't come back any thicker over here. It's a little more extensive, but the thickness is perfectly normal over here. So it's essentially an example of that sort of that ultra-sensitive behavior that you predict. Now, of course, hysteresis isn't just a bad thing. Hysteresis can be a good thing. If you want to have switch-like behavior, if you want to turn something on and off at different locations, if you can externally affect whether you're in this high p-value state or low p-value state, maybe that can be used in morphogenesis. Maybe that's one of the ways you get these structures is, say, for example, you ignite growth over here and you extinguish it over there, right, using these feedback factors. And so over the last couple of years, we've been involved in a collaboration with John Lowengrub at UC Irvine, building some fairly simplistic models, but models that have the right spatial geometry. And these are continuum models, but they take into account a lot of stuff a lot of non-uniform boundary conditions, and they just enable us to explore what you would get geometrically if you allow these positive and negative feedbacks to interact. And so here's some of the things that you see that you might expect. Here's a case in which cells are growing, but the positive feedback gain is high enough so that the thing just keeps expanding forever. The positive feedback always wins. You go down just slightly lower in the endogenous positive feedback gain, and the thing can't get off the lower branch and essentially extinguishes itself. The interesting case over here is you have the same parameters as over here, but you have a little bit of an external signal, a source that transiently adds the positive feedback factor over here. And you'll see what happens is the thing starts growing, and then it ignites over here. And then you take away that source, and it doesn't matter, because it's still self-sustaining. Like the psoriasis lesion, it's gotten beyond the point where you have enough stem cells that it will keep itself going. And as a result, you get very characteristic morphogenetic behaviors out of it. And if you go back to the olfactory epithelium, you say, well, where are these things that regulate the strength of FGF and GDF11? And you find that they're very spatially regulated. So for example, FOXG1 is high here, high there, low there, and low there. These markers of FGF signaling are high here and high there, low there, and so on. And those correlate very nicely with what's going on in the morphogenesis. And also they correlate with what cell types are present. So you see much higher numbers of these intermediate cells in just these places where the FOXG1 is low, and low numbers in the places where it's high. And that's what you would predict. In fact, you can go in and using these pulse chase experiments, you can actually map out the p-values. And you can see there are abrupt changes in p-values. So over here, where p is 1, the tissue is essentially doing exponential expansion. And then abruptly, it's switching to a place where it's at homeostasis. And it's hardly changing at all. And so if you can do that spatially with sharp transitions, obviously you can make a lot of really interesting morphogenetic structures. Okay. Now, earlier I said there was one more thing we had to explain, which was how is it that when you take away this feedback, these cells don't explode? But if there were any other feedback on here, then that would ruin the perfect robustness you get from having that feedback loop. So how do you get around that problem? And so we played around for a very long time looking for other feedbacks that will do, and we finally realized the problem was you don't need a redundant circuit. That is another feedback that acts in parallel. What you need is a fail-safe circuit, a feedback that doesn't kick in until the first one's broken. Okay? So how in the world would you get a fail-safe circuit in here? So it turns out there's a phenomenon that's been well known for a long time, and it's closely related to, I think, something Boris is going to talk about in his talk, which is the mechanical effects of cells on each other, on their rate of division. So essentially, when cells get crowded and squeeze on each other, right, and under compression, their growth rates, their division rates slow down. And this is observed almost universally. It's certainly observed in cells in culture, right? You can also observe it in tissues by squeezing them. And more recently, it's been suggested what the molecular mechanism is. If you're interested, it's not really important in this context, but there actually are transcription factors that move from being in the nucleus to being out of the nucleus, depending upon whether the cell is under compression or under tension, right? And how that's done is an interesting story. So the interesting thing about it, if you have this self-feedback of the cell on its own growth rate, right, then it can actually explain very nicely the data that we see in the case of the activin mutant. Because what happens when we take away activin, this p-value goes up, these numbers go up, but as a result, their rates go down of division, and so consequently, they don't contribute much to the next layer. And that's why you don't get changes at these layers when you get changes at that layer. You had a question? Well, for the ones that divide, obviously it doesn't matter here. But we don't actually know for sure for all the cells, right? Because in this case, we're still trying to figure out exactly which ones experience this, but it's a very common phenomenon in cells, so much so that it might just be universal in most cells. But we don't know is the short answer. Okay. Getting close to the end here. This whole discussion begs an interesting question, which is why would you ever evolve a fail-safe control, right? I mean, think about that. If by definition a fail-safe control is something that only kicks in after something else has broken, how would it ever evolve? How would it ever be under selective pressure unless that other thing is breaking all the time, right? And so we kind of puzzle about that because we didn't think that evolution really cared about activin knockouts. You know, much easier to just let them die than come up with a system to compensate for them, right? But what we realized is what's much more important to the cell, to the organism, than whether activin is knocked out body-wide, is whether each individual cell might undergo a change that changes its sensitivity to activin, right? So let's think about this. Imagine you have, progenitor cells that are producing terminal cells and feeding back in just the way I described. But now let's think about cell-to-cell variability. What happens if one cell picks up a mutation that just slightly changes its sensitivity to activin? So it needs more activin to be pushed back towards that p-value of 1/2. Right? Well, what's going to happen is that cell will continue to proliferate under conditions when these are held in check. In fact, it won't be brought into equilibrium or steady state until this number grows substantially larger. But under those conditions, the p-values of all these now drop below 1/2. And what happens? That means they extinguish. So essentially, cell competition is set up by the slightest change in sensitivity to the negative feedback. And that's a real problem, right? Because you'd always be selecting for things that are less sensitive to the feedback. And eventually, they'd be taking over. In fact, this does occur in special cases. There are a lot of cancers that show a phenomenon called field cancerization, where what you see clinically is that the cancer arises in multiple locations in the same tissue. They're not metastases. They're multiple primary locations. And what's happened is there's some pre-mutation that, because it's selectively advantageous, has swept through the tissue but didn't cause cancer. And then that sets all the cells up for a high rate of cancer development. Okay.
Yeah, but in this case, they are still going to feel the negative feedback due to crowding.
That's right. Okay, so I'm glad you brought that up, right? Because then the question is, what happens if we put that in, right? What happens if we add in the negative feedback due to crowding? Can that compensate for this phenomenon? And so you have to do stochastic simulations at this point because we're asking about what happens to individual rogue cells. And the interesting thing you see, so in this case, the mutation in the stochastic simulation, what it does is it makes the mutant cell have a p-value higher than 1/2. Okay? And what you'll see is sometimes those cells will indeed sweep through and take over all the other cells. Right? Sometimes they won't. They'll sweep up and then come back down again and be extinguished. And the puzzling thing to us was the higher we made the p-value, that is, the more that cell just does nothing but renew, the more likely it was to extinguish, which is kind of the opposite of what you'd expect, right? You'd expect normally that the less it renews, the more greater probability of extinguishing. But if you look at it closely, what's happening is, as the cell goes up in number, right, it's depriving the neighboring cells of the negative feedback they'd normally get because it's renewing all the time, so it's not making the differentiated cells. And so consequently, the normal neighboring cells also increase their p-value as a result of being deprived of negative feedback. So everybody locally is trying to divide faster, and that creates a huge pressure, right, which then slows everybody down. But the guy in the middle who started it off feels the worst of it, right, because he's right in the middle. And so his probability of being slowed down more, right, is greater. And so therefore, he gets a high rate of extinction, right? And the reason it depends upon having a very high p-value, right, is because if it doesn't, if it, say, has a p-value of like 0.6, so it still mostly is 50-50, but a little biased towards renewal, well then it still makes a lot of differentiated cells, and the neighbors, they don't get excited anymore, right? They still think they're in a normal tissue. So it turns out that works kind of on its own, right? Depending upon the growth rate of the mutant cell and the p-value that you give it, right? In regimes where the mutant cell is simply going to the strategy of divide all the time, those are the cases where it has the highest probability of the mutant cell extinguishing and the lowest probability that it fixes. So light means it doesn't fix. Okay. And that's interesting because that predicts a phenomenon that's observed and is very puzzling in the cancer world, which is that most cancers do not consist of a single cell type that just divides. Most cancers, in fact, have lineages. Right? They call this the cancer stem cell hypothesis, but they really should call it the cancer lineage hypothesis. Because what it is, the cancer cells are dividing and producing differentiated progeny, which can't divide anymore. They're not cancer forming. It's only a tiny fraction of the cells in a tumor are sometimes the only cells that can go on and make new tumors. People are very surprised by this, but in fact this is an observation over a century old. Right? That's how we diagnose cancers, is by the differentiated cells they make, which in most cases are usually readily recognizable. This result actually predicts that that's an advantage to the cancer cell. It essentially enables the cancer cell to be less detectable by the normal cells around it because it's still producing the negative feedback and not depriving the normal cells of it. Okay, so a lot of questions still remain about what I'm loosely calling proliferative dynamics, right? Understanding how proliferation takes place in lineages and how is it controlled. For one thing, we don't actually know the cellular machinery that makes these changes in p-values. We have some guesses. We know, some of the things at the beginning, the growth factors and so on, but exactly what that does to the cell to change the probability of renewing versus differentiating is still very poorly understood. We don't know too much about how lineage progression controls the accumulation of mutation, although there's another story I didn't have time to tell you about that suggests that this high degree of symmetric division actually plays a positive role here. And we don't really know how important proliferative control is in morphogenesis, because there are other mechanisms by which morphogenesis also takes place involving, say, cell movements, cells pulling on each other and mechanically changing and so on. So lots still to be understood. But in terms of general lessons to take home, first thing, remember that these interactions that control growth, regeneration, and morphogenesis always play out within lineages, right, in animals. Right? We're not E. coli. None of our tissues are actually just a single cell type that's growing up to a certain point. They're always through transitions in lineages, right? The behaviors of stem cells and these intermediate cells are not cell type behaviors. They're just emergent properties of feedback circuits. You could take the same stem cell, put it in another environment, and it might behave like an intermediate cell or a terminal cell, or it might divide forever. In fact, that's why we can culture stem cells, is you can convince them to divide forever. Regulating the speed of the cell cycle, counterintuitively, is not a particularly useful thing to do. Right? You would think that that would be the first place you would regulate if you wanted to regulate growth, is slow or speed up the cell cycle. But it turns out because growth is an exponential process, regulating these transitions from one lineage stage to another is much more powerful way of controlling things and is the only way to get at perfect robustness or integral control. And finally, the systems that control growth are intertwined with the gene regulatory networks that control cell fate. And that's kind of related to this point of things happen within lineages. So by controlling the transition that affects the sizes of things, you're also affecting which cell types you're making and in what proportions. So the tissue has to solve all these problems simultaneously. Okay, so I'll just thank some of the people who've been involved in this. All the stuff I told you about the olfactory epithelium is part of a long-term collaboration with Ann Kaloff's lab, who really pioneered the use of this system something like over 20 years ago now. A lot of the math is collaboration with Ching Ni and his group and John Lowengrub and their group. And these are some of the people in my lab who've been working on this particular problem. Okay, thanks a lot, and I'll take any questions you have. Yeah. Plant tissues, very different. There are stem cells and there are lineages, but there tends to be much less regulation of size, right? Plants tend to have lots of cases of fairly indeterminate size. The leaf could just get bigger and bigger and bigger. So in addition, the signals that can be passed back and forth between one cell and another are often very, very different in plants and are flowing in, through transporters from one cell to the next. So to my knowledge, a lot less is known about these issues of growth control in plants, but some of that may just be my ignorance that I don't know who's working on it right now. Any other question?
So in one of the performance objectives, you mentioned that there should be stability in the lineages. So for that, there is extra negative feedback on the intermediate cells. So has anybody shown that removing such extra, that extra negative feedback gives oscillation, instability?
So that's a really interesting question. The short answer is not quite, because the number of cases in which we actually know the identities of the factors and in which we can also follow the dynamics of regeneration closely are sort of non-overlapping. We know the factors best in places like the olfactory epithelium, and to actually do dynamics of regeneration experiments is very expensive and time-consuming. But in the hematopoietic system, where we don't know the names of the factors very well, but there's very good evidence that the factors are working the same way, there are a number of examples of induced oscillatory behavior. In fact, it's very common in various myeloproliferative syndromes, in leukemia, in autoimmune syndromes, to see this month-long oscillations, which fit, because these are relaxation oscillations, where the relaxation time is set by the death of the extra cells that you're making. And many people have tried to model hematological oscillations. And of course, as oscillations, you can model them lots of ways and fit the data. It doesn't really help. But so you certainly do observe such oscillations in vivo. In the olfactory epithelium, if you look at the little bit of dynamic data out there, you can see a teeny bit of oscillation in the wild type case. And the same thing in the liver. When you do major liver regeneration, you can actually see some damped oscillation. But what you're saying is you'd make certain predictions about could you actually enhance those oscillations by taking away certain feedback loops, and it just hasn't been done yet. Yes. So I wish I had another hour. Yeah, so you pointed out a really important thing, which is, I told you all about ODE models in which we assume that these feedback factors are well stirred. And of course, that's nonsense. They're not well stirred. And these tissues stratify so that all these cell types are over here and those cell types are over there. And so in fact, these molecules are acting in gradients. And they have the same parameters associated with them, like length scales. How far away can it act? One of the consequences of recognizing that is realizing that you can coordinate growth, but over only a certain spatial range with these factors. Another thing that I don't know if I should, well, I don't have time to show you another slide, but it turns out that stratification is really important. So in the case of the olfactory epithelium, the stem cells are all at one end of the layer of cells. They're what's called the basement membrane, the bottom layer. That's a permeable layer. Anything can flow right through it. And right at the other end of that is a huge concentration of a molecule called follistatin. And what follistatin is an irreversible binder of GDF11 and activin. So essentially you have a perfect sink sitting just under the epithelium, and the stem cells are all right next to it. And what you can show is that's the optimal solution for extending the range of size of the epithelium over which the stem cells still have good sensitivity to that size. Because essentially what it does is it takes that gradient and it makes it almost a straight line gradient going down to the epithelium. And so as it goes bigger and bigger, the slope of that line changes over many length scales instead of what you'd normally have, which is boundary effects only occurring over about one length scale. So yes, these spatial things matter a great deal. And in some of that stuff I showed you of sort of tissues and morphogenesis, of course, all that's dependent on the fact that these morphogens don't just diffuse everywhere.
What about flare-ups in oscillations?
Sorry, what are?
What about flare-ups in the oscillations?
Flare-ups?
Yeah. So in real situations, what happens that the oscillations in immunological disorders, that it will come down, but not in the previous state. It will come down to a certain extent, then again it will oscillating from that state.
Yes.
So what about that?
Well, I know of a model. I mean, something must be changing over time, right? But I know of a nice model of oscillatory responses in the non-obese diabetic rat, in which the T cells eventually kill the pancreas, right? Because there's a particular antigen. Yeah. And they oscillate in this model. This comes from Leah Keshet, a mathematician at UBC. And they oscillate in this model for what turns out to be exactly the same dynamics that we described. It's that same effect on p-values. They just have different terminology. And there, in the model, that same thing emerges, right? Which is the first oscillation doesn't go to baseline, then another, and then eventually, after the third oscillation, you kill the pancreas and you're dead, right? Yeah. So I'm not sure exactly. I mean, I could look it up, but I'm not sure exactly what the mechanism is to explain why you don't keep coming to baseline.
Okay. So, you presented your lecture as a series of problems and then solution, generating another problem,
right.
another solution, and so on, right? Now, so in an evolutionary context, how does one expect this kind of complicated problem solving
yeah.
have arisen? I mean,
that's a great question. No, I can't really give you a very intelligent answer to that, because the basics of these circuits go back to the first epithelia. They go back so far in evolution that it's hard for us to to bring to bear any any sense of what evolved first, right? The epithelium, in many ways, is the most amazing evolutionary invention. And if you look at the evolutionary record, right, it just it's suddenly there, right? And the cells are all arranged in a row, and there's a basement membrane, and there's polarity. So all these things are there as at once, and we have no idea how it all evolved. But control of growth had to be part of that. And if you just think about an epithelium, if all the cells are individual actors and there's any slop at all in how they're behaving, their cell cycle rate, in their p-values and so on, you can't even keep the epithelia flat, right? They'll get all bumpy and out of control. And so all I can say is I know these are ancient problems that had to have been solved very early on, and that makes it difficult for us to even guess at what the trajectories were that enabled them to, the evolutionary trajectories were. And maybe Boris will talk more about that. Are you going to talk about these kinds of things? Yeah, okay.
but given the design that you presently have,
mm-hmm.
I mean, from the structure of the design,
mm-hmm.
is it possible to at least sort of put a kind of an educated guess that this must have come first,
yeah.
and that must have come later in the design?
You could, but I would personally shy away from it. I feel that there's heuristic value in presenting it this way and saying, well, here's this problem, and then this solved that problem, and then this solved that problem, but mainly because it helps me understand. I'm reluctant to say, well, and therefore I actually think they got solved in that order, because my intuition tells me that I'm going to guess wrong a lot, right? So, but I'm happy to be in that position and say, well, that's okay, because really I'm just using this as a way to understand essentially how to modularize the system into modules that do different things. But it may not have, evolved that way. Okay?
Okay,
I think
so
we're like a break.
then we break and we come back in at 11. There's tea, coffee outside.